Hello, everyone. Welcome in today's lunch talk. As always, I will give everyone the time to join us before we get started with the real talk. And I will start with a short introduction of our SciComm Academy. So my name is Annelies van Dijk, and I am the project manager of the SciComm Academy at SciMingo. And for the people who might join us for the first time, SciMingo is a Belgian nonprofit organization who's on a mission to help people take their first steps in science communication. And specifically with our SciComm Academy, we organize, for example, hands-on trainings and workshops to help researchers share their work with a broader audience. For example, we have training courses in presenting on stage, in infographics, in popular science writing, etc. You can find all of our trainings on our website or by scanning this QR code, what will take you directly to the website, or you can subscribe for our newsletter using this QR code, and then you will stay up to date whenever we launch a new training course. For our next training course, which is next week on the 7th of May, is our Dutch popular science writing course, and there's still one spot available. So if you're interested in joining this, take a look for sure on the website or in the follow-up mail where I will share the link to this. Next to the training courses and the workshops, we also organize online lunch talks, as the one of today, about science or science communication in general, and we invite experts or science communicators to discuss these topics with us. Next up, on the 16th of May, will be a Dutch panel discussion on how to write a scientific bestseller or a popular scientific book. So we will invite various authors from different fields and to share their experience and to answer all of your questions. So if you're ever doubting about writing a book about your own research, it's certainly worth joining. And for this one as well, I will share the link in the follow-up mail or in the next newsletter. So let's see. Yes, a lot of people are joining already. So that brings us to the lunch talk of today, of course, where we will discuss art and science and how these two disciplines are actually not as different as they might seem at first or as we might think, and how art can actually help us or how art is a great tool in science communication. And this lunch talk is co-hosted with the Jonge Academie, which is an interdisciplinary team of scientists and artists that actually want to build bridges between these two disciplines. But I will not give too much information about this. I will let this up to the experts. So I will now give the word to Nicolas Bijens, who is a member of the Jonge Academie, and he will share a bit more about their mission, and I think also about the hurdles that come along with building these bridges between the two disciplines. I still want to end by saying that it's, of course, an interactive discussion as always. So please ask all of your questions via the Q&A window or via the chat, and we will try to tackle them along the way or after the various speakers have shared their experiences. Nicolas, the floor is all yours.
You mentioned Nicolas Bijens, and I'm here as a member of the Jonge Academie and as an artistic researcher at the Royal Academy of Fine Art in Antwerpen. We are an organization that brings together young, or let's say relatively young scientists and artists, and we believe that innovation often arises when different worlds collide. For us, interdisciplinarity and even transdisciplinarity is essential. It's not about disciplines working side by side, truly thinking, creating, and communicating together. So why do we work on both science and art? Because we engage with pressing social and environmental issues, often with a strong focus on education, and we believe that these problems deserve to be explored for multiple perspectives. Not just solved, but questioned, challenged, and reimagined. But then how do we work on science communication and art together? At the Academy, members can join working groups based on their interests. Not all groups are perfectly balanced between artists and scientists, but I will give you a few strong examples. Of course, I'm here also as an artist, so the projects where I'm involved with are more artistic than only scientific, so it's a mingle a little bit. Of course, we are running now at Earth, and that's a group focused on sustainability and ecology. It's preparing an artistic research project around unforeseen and climate change, with a special focus on Iceland and melting glaciers. In 2025-2026, we will launch a small international artist residence exchange. 3 Icelandic and 3 Belgian artists will work together, selected via an open call. We want to connect the urban landscape of Brussels with the natural landscapes of Reykjavik and Iceland, and use art as a way to rethink global ecological issues. Then in 2023, with Annelies van Assen, I did artwork. It looked at the development of young artists. How does an emerging artist grow into a sustainable professional practice? In the port building in Antwerp, we organized 4 days of dialogue and exchange involving galleries, commissioners, media professionals, policymakers, curators, and of course, artists themselves. Another small project we did, what was not so small, was the Bustebende. And it addressed the representation in public space, particularly the image of artists. At the place of Academey in Brussels, only statues of old white men were or are still on display. And then together with the Royal Academy of Fine Arts in Antwerp, we created new works that brought more diversity to that space. We didn't remove the old statues, but we complemented them with new ones, made by art students, by the way. So that was quite a nice collaboration. And finally, of course, every year we bring out the MaJa. This is our magazine, and that's the place where science and art meet each other in print, with contribution that challenge conventions and offer. So, but what about the admission and the diversity in the Jonge Academie? Initially, artists applying to the Jonge Academie were asked to show affinity with science, and today we ask the same of scientists. So they have to show affinity with the art, so that there is a more kind of collaboration. We deliberately break with compartmentalization. We work across disciplines, across universities, and across cultural boundaries. But of course, there are also some challenges and tensions, not always so smooth. It isn't always easy. Science and art often remain separate in practice. People tend to stick to what they know and to their own practice, timeline, and tools. From within science, we see different ways of thinking. That's quite clear, right? The humanities scholars often align more closely with artists. And then the natural scientists, surprisingly, can be very open to artistic collaboration, precisely because art is not part or not the main part of their daily research routine. In our work on science communication, we are very aware of one risk, that art is reduced to a tool for communication. So communicating science in a visual aid or an illustrative layer, and that's what we try to avoid. We believe in something deeper. Artists are not the illustrator of scientists, but they work together. And they are authors, thinkers, and makers in their own right, opening new perspectives together with scientists, not after them. There are some things that are coming up, some concrete examples of synergy. Next summer, we will participate in Theater aan Zee in Ostende. I think everybody knows it. Usually, science appears at science festivals, but here we brought science into the cultural festival on equal terms. We organize talks and performances that translated complex research into accessible formats, but always with care and not lose depth. That's also, of course, the danger. The challenge, how to communicate clearly without simplifying. Then there's another thing that we always involved in the PhD Cup, but of course PhDs also exists now like artistic doctorate, and the Academy is always part of the jury of the PhD Cup, National Science Communication Competition, and we are seeing more and more interest in artistic perspectives, but it raises, of course, new questions. When and how can artists with artistic-based PhDs enter traditional academic settings? How do we deal with artistic research in systems that often prioritize objectivity, measurement, and peer review? So to conclude, working across disciplines is not always smooth, but it's necessary. Our goal is not just to translate scientific findings into artistic forms or vice versa. It's to challenge assumptions, deepen perspectives, and enrich our understanding of complex problems. Together, artists and scientists can open up new spaces beyond the binary logic of true and false, beyond simple narratives of progress. And to end up, here's our core. Science and art are not extensions of one another, but can meet in the formulation of new questions and new insights. So thank you very much for listening.
Thank you, Nicola. I maybe already had a question because you were talking about the open calls and for the projects. If people are interested or think they're the ideal fit for this or future calls, is there like a newsletter they can subscribe to, or how can they stay up to date?
I don't know if you have a newsletter, but they can always check the website. We're also now constructing a new website with a new layout, and it will appear soon, I think, one of these months. And there you can just follow all the projects and all what is going on. Okay, I will for sure link the website as well in the follow-up mail, then people can stay up to date and look out for the launch of the new website. Thank you very much. And of course, if there are additional questions, please drop them in the Q&A or the chat. And we will, in the meantime, continue with our next speaker, which are two speakers at the same time. So we have Pascal Pollier, which is a visual artist that's working on the connection also between art and science, together with Anne van de Velde, or Dr. Anne van de Velde, who is both a medical doctor and an artist herself. And they will share with us their BIOMAB project. And BIOMAB stands for, if I'm correct, Biological and Medical Arts. And it's a project from the University of Antwerp. And they want to use art to help science communication, but also medical education. But I will for sure let them introduce it even better themselves. So Pascal and Anne, the floor is all yours. And I think you will share a presentation with us as well.
Thank you. Thank you very much, Annelies and SciComm for inviting us. And to our audience, thank you very much for being here. The first slide is exploring the role of art in science communication. So what you can see here is a dissection of a cow's uterus, and the calf, a little bull, was only 3.5 months old. We saw this a couple of years ago during one of our BIOMAB dissection drawing days. It was pretty amazing. Not only is it very beautiful, but also very interesting to see, because we had no idea that a cow had 100 placentas, for example. So I can imagine that Andreas Vesalius had a similar sense of awe and wonder when he looked for the first time inside of the human body. And in a way, in those days, they used to describe it in a poetic language, which you can read in the books, the Humana Corporis Fabrica, on the structure and the fabric of the human body. Next slide, please. In our modern times, scientific descriptive language has become quite dry. But the artist Johannes Le Frank, which was from 1729 till 1812, was an 18th century painter, scientist, physician, and poet from Leiden, Holland. And he made these pictures some 200 years ago. And he describes these placentas as very beautiful and with a sense of awe when he observed them. And this can now be considered as poetry with an endearment. So figure 3 is a perfect lifelike image of an internal form, the funnel cavity with the piercings like mulberry with their nipples, everything pictured during natural secretion of the berries of a cow's womb. So it's so visual, and art and science is not just data, but a living, breathing experience. And it's not every day that you hear about the berry secretion and cows' wombs, right? So the next slide.
Yeah. Yeah, the medical arts history goes way back, and I've put here two examples of important artists working in medicine. Everybody knows, of course, Leonardo da Vinci. He has a huge work done. He is the ultimate combo of the artist and the scientists. And as Nicola said, it's so important that there is the synergy, the working together. We have to keep that during all these centuries past and continue that for the future. We also see Frank Netter, who is a long way after Leonardo da Vinci. And his books are still very used in modern medicine because he captures a lot of important diseases, very visual, much better than with only photographs or texts. So here you see two examples where art and medicine, they have always walked hand in hand. And the art of medical illustration, medical art, is still evolving. We have the European Congress, the European Meeting of Medical and Scientific Illustrators. It already has a long history. And here you see the team coming together at the University of Antwerp for the annual meeting. So as I said, the organization was founded in France in 1986. And you can go to their websites. You see what medical art, medical illustration, scientific illustration is all about. It's not only illustrating texts, but it's also thinking about how a scientific problem can be made understandable to the general public. And the general public can be a scientist, a baker, a teacher. So everybody should understand what science is about. And that's the main goal of the profession as a medical illustrator. And then to go back into history and to come to explain future slides, we all know all these beautiful paintings of anatomical lessons. And this is an example in the St. Jan's Hospital in Bruges. And you see in the 17th century, not only medical doctors, not only researchers, but everybody was welcome to attend an open dissection and to wonder about the beauty of the human body. And that's so important that we also can keep that in modern days, that the inside of the human body is communicatable to everyone, not only to people working in the field.
I should have had a little trigger warning in that we're going to have some sensitive images, but there's only a few, so and they're quite beautiful. So in Belgium, we have BIOMAB, Biological and Medical Art, and internationally there's ARSIC, connecting artists who dive deep into medical and scientific research. ARSIC encourages interdisciplinary cross-pollinating collaborations between artists, anatomists, medical students, and medical art students. We blend knowledge, technique, and creativity. With BIOMAB, we organize dissection drawing classes, as you can see here. So there's we do this as a weekend. One day is human anatomy, and the next day animal anatomy, which is comparative anatomy. And during these sessions, participants can draw or question or even try their hand at dissecting or learning surgical skills such as suturing. So it's very important for us to learn directly from observations, not just copying other people's work and illustrations, but really see for ourselves. And then also understand the functionality of these anatomical structures. As these are fresh frozen specimens, they can move if we pull on certain tendons. The body still moves as a locomotive system as in real life. Next slide.
Yeah, so this is an example on how students, medical students and art students, they attend these dissection classes. And when medical students sketch, they truly understand the complexity of anatomy. And that's always better than only the photographs. And the next slide, you see how these lectures are given. So at the forefront, there is the anatomist who explains the inside of the body. So you see here Professor van Glabbeek explaining the muscles of the upper arm. And the image in the far you see is from Bidlo, a very famous artist of the, I think, the 18th century. 17th, 18th century. And then there is always the link between the drawings of the past and the dissection in the class. And you see these beautiful images. The left one is by Luna Lucas, who is a graphic artist, a student in graphic art. And she made some really fine sketches during the dissections, which is not easy to do because we see that during these dissections, everybody is really fascinated and your eyes want to capture before you want to draw it. So, but then after 1 hour, people really start drawing to see what they see and what they want to explain and communicate. And then it ends in beautiful anatomical fine drawings like the feet next to it. And that's made by Pascal herself. So really good images of what medical art is about.
In 2007, we organized a conference, Anne and myself, for the AIMS called Confronting Mortality with Art and Science. And as part of the conference, there was an exhibition connected to it. And my resulting wax sculpture called Confronting Mortality was a collaboration between anatomist and orthopedic surgeon Francis van Glabbeek. He's also an elbow specialist and BIOMAB founder at the University of Antwerp. And the sculpture depicts a figure observing his own biceps. He's seated on 2 books of Vesalius's masterpiece, De Fabrica, and in a way he's observing his own mortality. In 2006, I completed a course in facial reconstruction in Maastricht together with my colleague Richard Neve and the colleagues from Maastricht University who are doing a medical and biological art course there. We just thought it would be wonderful to build the facial reconstruction of a famous person. And then we thought, who would be better than reconstructing the head of Andreas Vesalius, because he was such an inspiration to our profession. So the facial reconstruction course in Maastricht, afterwards, we just thought Vesalius himself would have absolutely loved this, as he was the first one to put the whole human body into pictures and describe the inside. But where was he buried? Where was his skull? And this sparked an entire new project, which is the search of the grave of Andreas Vesalius, but that's a whole other story. Understanding the muscles of facial expression is also crucial for both doctors and artists, the whole discipline. So I give this workshop, the anatomy of facial expression, and I will actually be giving it next week in Belgium, in Ghent. In order to express emotions in a work of art, it's very important to know what the anatomy behind it is. And speaking of Vesalius, he was a pioneer in linking medicine with illustration. His work revolutionized how anatomy was taught. My colleague and facial reconstruction expert Richard Neve and myself made this portrait in bronze, which is a half portrait, half reconstruction. to sponsor our 2014 monument, because in 2014 it was the 500th anniversary of the birth of Andreas Vesalius. So this head was made in anticipation of finding the skull, and we worked from the outside inwards, from known Vesalius portraits, and we imagined what his face might have looked like. So this is the monument we funded with the sale of these previous sculptures, and the monument portrays the écorché based on two of the écorchés in the Fabrica. He's holding a skull symbolizing the quest for the grave. Ricochet, Vesalius's life, work, and creation is contemplating and looking at the skull, which symbolizes the search for the grave. We still haven't found his grave. It is on Zakynthos, but we are much closer now. Please follow us, follow BIOMAB and ARSIC for more information about that in the future. Our brains, the ultimate frontier, left and right side of the brain symbolizing here art and science, the perfect marriage in the two hemispheres of the brain. Medical art also sheds light on microscopic level. The pigment cell I made as part of my albinism project brings the invisible world into view, or the minuscule world. The beauty, there's a great beauty, a world within a world, in the hidden, in the unseen. So medical art can show us this unseen and beauty inside of our body and the systems we rarely think about, but depend on every second. You might not think about your cells working away in the background, but they are keeping you alive at every second.
So the last part of our talk is about how to integrate the visual arts again into the medical and also the scientific curriculum. Many centuries ago, there was really a huge link between the arts and the scientific world, but they grow a little bit further away, and now they are coming back again together. And this is an example of a study master students did. And they interviewed bachelor students at the University of Antwerp in the medical curriculum. And they wanted to see which illustration a student found most valuable to have it in their studying anatomy. And they used all kinds of different illustrations, going from the past to the current illustrations. And the conclusion of their thesis was that by exploring more visual work, not only literary work, but illustrations, artworks, it can deepen a student's understanding of anatomy. But by using the arts into the curriculum, it enhances also communication skills, and it can foster creativity. So actually, we really wanted to have the students have active drawing lessons in the curriculum again, but that takes a little bit of time. But I think in the future it will go like that. So it was a bit of a passive work, but in the future we really want to have the drawing classes back into the curriculum again. And the interviews, they put some quotes on it, and it's absolutely true, of course, that when you draw things yourself, they become more firmly anchored in your brain. There is a connection between the hand that does the drawing and the thinking, because you're actively engaged with them. So it's really important to highlight it in this highly digital world. And another project done by students in the medical curriculum is if arts can help as a language of health in a doctor-patient conversation, because we know there are a lot of informed consent formulas, sometimes 30 pages, and the visual word is not often used enough. And we speak a lot of health literacy. We know that it doesn't depend on the degree you have if you understand your body quite good, and a visualization of it can give a more inclusive knowledge of your health problem. And then to finalize, I think it is important to show this image. It's an image we were invited to make by the group of the health humanities at the Antwerp University, the Metrodora group, and they have a lot of interdisciplinary meetings. And this is two hands. One hand is of the artists, one hand is of the scientists, and it is now permanent exhibited in the foyer at Drie Eiken campus, so the science campus, where there needs a lot more integration of art. It is inspired by of course, of the sculpture that we see at Grand Plaza in Antwerp, where the Brabo cuts off the hand of the giant, and it's on the viewer who will decide if the giant hand is of the artist or if the giant hand is of the scientist. So it makes a connection, and it makes a big communication when you have when you are in the foyer having a drink with colleagues. So of course we cannot do that without a team. This is the members who are most involved in the BIOMAB projects. And you see that art makes science human, and art makes knowledge unforgettable. And I think it's very important to highlight that. And if you want to read more on the use of art in science communication, this is a very good book. It's part of a whole kind of books published by Springer. We were invited to have a chapter in it, and you can use it if you are a scientist seeking to enhance your ability to communicate your research, or if you are an artist interested in biomedical visualization. And I think with that, we close our lecture, and we are happy to take questions.
I can ask. This is all about medical education and medical science, and it's very relevant. And there's also the international group. Do you know if there are similar things connecting other scientific fields with art in Belgium or in Europe? And do you have like a good connection with those initiatives as well?
Yeah, Pascal, you can.
Yes, yeah. There's a whole network. There's a bio artists, sci artists, so which is a bit more broad. It's not just anatomical sciences, but it's all sciences and all art forms. And we try with BIOMAB also to support these groups. But yes, we do have a very big international network of people that stretch as far as Australia, Symbiotica in Australia, yes, in America, in Europe, everywhere. So we do have connections with those.
And I think also, of course, the initiatives that Nicolas said at the beginning are very relevant here as well. And I think there might be a lot of overlap in between. Maybe Anna, I also had a question for you, because you shared a nice quote of one of the students who said that drawing yourselves helps with studying and reminiscing things. So what is then your vision on AI and AI generated art, but also in general AI for medical education and communication? Because I don't think AI is going anywhere, and also AI generated art is here to stay. So I'm very curious what you both think of this.
Yes, actually, yes, you're right. It will stay, and it is used, and it is a hot topic. It's also the question students in arts education on the sciences, because I can add to what Pascal said. We are actually involved with a group of Maastricht who have a course on 2 years. It's a master on scientific illustrations. And it's much more than only medical illustration, but it's all sciences. And when we have a meeting, when we have a European meeting, and I can imagine an international meeting, the students always want AI on the topic for presentations. So they are really involved in it, actually, to learn more and to see how their future, because of course their profession will be artistic reflections, and they want to know how their profession will change when AI comes into it, because you hear the feelings that scientists will use more and more AI generated images for their communicating their work. And of course, it is not only AI that can solve that. So we need to stay in communication with the visual artists for the moment. And I think Pascal wants to say already something.
Yes, we recently, our last MA and AIMS joint conference, which are the medical artist associations of the UK and Europe, was in London, and the main topic was AI, and everybody was really, really interested in it. And there are conflicting opinions about using AI. But I see it very much, AI is our friend. You know, we use it as a tool for creating certain things which, you know, we already use certain tools in Photoshop and Illustrator and in 3D illustrating programs. To a certain extent, automation is already there. So if you want to use it as a tool, it's excellent, because it can generate some things that can be inspirational. But if you are thinking of that it will replace our job, no, it can't, because you can see that, you know, anatomy in AI is not correct. So we and also the concept of certain illustrations really require an awful lot of research and understanding that I don't think AI can touch upon yet. So there's a whole lot more than just making illustrations. You know, there's the understanding of the concept and research behind it in order to communicate it. So it's a long way off taking our jobs anyway.
Yeah, I think it's more indeed, it's a very complex thing, but as you said, in general, if you see it as a tool, that's a good way of looking at it, probably. There's a few other questions that came in. One of them is Pascal, if maybe you can share the links to the BioArt and the SciArt groups. If you share them in the chat or to me, I will share them for sure in the follow-up mail as well. And then someone asks if you can say something about other art forms than painting or drawing or sculptures. What about music, poetry, or acting? Is there also groups or initiatives connecting these with science? Maybe also Nicola and MaJa, feel free to answer if you are aware of this.
Yes, maybe I can just quickly answer. We do have exhibitions, fine art exhibitions connected to our conferences with BIOMAB and ARSIC. So we do always include poets, musicians. So, you know, it's all the arts and all the sciences that we support with BIOMAB. So definitely, yeah. So if you look at the ARSIC website, which I will share later on with you, because I am not on the other side. I'm just got my iPad, but I will share the links to that.
Yeah, I can add very short also, because I work in hematology, so in the blood diseases, and we had a very beautiful project, a collaboration on the bone marrow transplantation, and it was really a great project because the acceptor for the transplant was a musician, and the donor was a performance, a theater maker, and they made a play involving dancing and visual arts, and they performed that for the general public, they performed that for hematologists, they performed that for stem cell transplantation acceptors, so all the fields, and it was really made the very difficult theme of a transplantation very acceptable for all kind of people. So it's a really good question, and not only the visual arts, but involving all kinds of art forms. Really important.
Nicola, you also wanted to share something, I think.
Well, I put the link in the chat. So I was thinking about Forum Plus. That's a magazine that is published by the University of Antwerpen. And it's a collaboration between the university, but also the Conservatory and the Royal Academy. So the Conservatory, that's of course about dance, theater, and music. So there you have more art forms that comes together and works around science communication and artistic research. So I think, but I put the link in the...
Yeah, I shared it with everyone. Thank you. Okay, I think that it's time now for our last speaker today, which is Dr. Maria Botto. Maria has on one side a PhD in life science, but also a lot of experience in the field of bioart. Currently, she's also a member of the Jonge Academie, and she's a researcher at the Royal Academy of Fine Arts and the Conservatory. And today she will explain her work on sustainable color use, but also tell us more about the laboratory, which is an experimental lab where artists and scientists can actually collaborate. So with that, Maria, the floor is all yours.
Okay, yeah, first of all, thanks for inviting me. Thanks for letting me participate in this talk, especially because my work is not so focused on scientific communication, but very much into the fields of art and science. So as you said, I'm trained as a scientist. My PhD was from the pharmacy school, so nothing related to the arts. But after, when I went into the postdoc world, I was looking for something a bit more creative. And then I was curious about many things, including scientific communication. Back then I had the opportunity to work as a lab technician at the Bach Society in Amsterdam. That in fact is a kind of a funny place because it's presented in one of the most famous Rembrandt paintings they have in this building, the Anatomical Theater. And the anatomical lesson of Rembrandt, it was part of is represented on what was there. And while being there, I was just simply a scientist helping out artists and designers to more find their way in the scientific world than actually doing something with them. Because we had a lab, it was a small lab, but it was a bit more like, okay, receiving people with the ideas like, okay, I want to become an algae, how can you help me with that? And then it was more about, okay, let's discuss about photosynthetic cells or photosynthesis before thinking on how you can become an algae yourself. And then in the Netherlands, the network of bioart and biodesign is quite developed, and it was quite easy to find connections within the scientific faculties and institutions for these artists to access and to work with them. And while being there, I was approached by the person who at that time was the head of design to create something similar here in Ghent. So the idea was to build up a kind of lab, bring tools and methods more connected to the scientific world and place them in an art academy to see how the students could work. And that's what we did. That's where I'm working today at CASC, is the Royal Academy of Fine Arts. And within CASC, I'm running the lab. So today I will show you my two jobs somehow, although they are very much connected. On one hand, I'm the responsible for the lab. And on the other hand, I'm the main researcher of a research line based on color production and application. Sustainable color production and application that can be applied in the fields of art and design. So the lab is named Laboratorium. At the beginning, when we had this idea of creating a lab, it was not funding for infrastructure. So we had to find another ways to get the funding, and then we got a research project. And the way that we thought, okay, how can we do research and at the same create a lab was by building up a lab on the topic of color, because somehow, color is connected to all the disciplines here within the school, and also because of the ecological impact of color. So many I mean, the production of color and the color waste management and the application of color has a huge ecological impact. And we thought that it would be nice to bring to the school other possibilities of making color and applying coloring in their works. Nowadays, the lab is much bigger and nicer. At the beginning, as I say, was a research project, and most of the devices that we had were second-hand scientific devices from other institutions. But yeah, nowadays we got some extra funding to create a proper infrastructure, still with many restrictions, but because we are in an art academy, so I cannot do many things that if I would be in another type of institution I could, but good enough. And in the lab, if you come to the lab, you will see all these scientific devices, and they all have protocols, and also there are some basic protocols to work with different types of materials that are the most common materials here in the lab, like working with algae or bacteria or fungi. As I say, all the materials that are entering in the lab are safe and controlled by the safety department. And in the lab is also used in education. So there are workshops. I teach one semester on color for master students. So during 4 hours a week, they come, and every week is one type of material and one type of scientific, or one type of artistic application. Like one day is microalgae and then screen printing. Another day is bacteria and seaborne dyeing. And then we have this website where you can see the colors that we are using, the projects that our students are doing, the activities that we are organizing, also an Instagram account where you can follow up on the things that we are doing. And here in the lab, many students pass by. The only thing that that is quite a challenge is that, as you all know, because I guess there are many scientists in the room, scientific research takes a while. And many times the art students, they don't have too much time. So the projects are a bit limited on time, but some students that they are really engaging with the material or with the technique, they can be here for maybe 2 years while their master doing experiments in the lab. And yeah, the experiments are quite broad, although most of the times somehow is connected to the color research, but we have students working with the visuals like microscopes, with creating perfume, working with chemical reactions for making movies, for making another movie about biology, this with crystals, making also performances, objects, photography, using mushroom, slime molds, coloring textile with bacteria, working with bees. So it's quite broad, the amount of topics that the students are doing in the lab. But as I said, many times it's connected to the color research. And now I will pass to the research that we are doing here in the lab. So at the beginning, when the lab was formed, it was mainly about pigments. We were working around ways of obtaining colors and pigments in a sustainable way. So the first part of the research was looking into natural materials and traditional coloring techniques and what was out there and how we could bring it back into the school, and working with the students with the pigments and also trying to understand why they were not longer in use. We created a big database of many plants that you could find and different type of shades that you can get, mainly to be applied on paper and textile, but also, we did some experiments with 3D materials. These are some of the comments that the students gave us, like why we are not using sustainable or plants because of the time, the price, the lack of the hard of reproducing the same colors. And after this first part, we went into more biotech, looking for microorganisms that could provide a color and that we could use and that we could cultivate in the lab and extracting the pigment and applying the pigment. The first part was we were working with bacteria, but we had some limitations in the type of bacteria that we were able to use within the lab. And afterwards, we passed to microalgae, and with microalgae, everything was smoother in terms of safety. And we also found out that you can get many colors with microalgae. Also, in this process, we collaborated with the University of Ghent, because just by a coincidence, we saw that the same algae that I was using to extract pigment and to make paint out of it, they were using for cleaning water. So we published a couple of papers on how you can clean water and at the same time obtain the pigments from the algae that are growing in the wastewater. That is a type of material that afterwards you can only use it for biogas or biodiesel. Normally what we do in the lab is the more scientific research is done in the lab, but we are always open to collaborate with external partners and with the students. So if the students come into the lab, they can use our materials and our techniques. And also if the external partner wants to join the project, is always welcome. Normally the deal is that the project has to be something that will add some knowledge to our own research, so that they will try a different type of material or they will test other characteristics that we didn't test. So we all learn about the process. And we have been doing the collaborations with a bookmaker making covers with microalgae. So what you see there is microalgae pigments on several books. We have been also doing 3D filament with polylactic acid and microalgae pigments. Polylactic acid is a kind of biomaterial, and you can color it and you can make film out of it. We also have been testing the colors on different type of textile and testing in a weather chamber how pigments react. Also making tests on digital printing with another student from another university, and also working with other artists, external artists like Amandine. She made this huge carpet colored with microalgae. And since 2020, the project of with the pigments ended because of a lucky encounter with a scientist. So I was in a conference in Brussels around color, and I presented the research on pigments with microalgae and bacteria. And he told me, like, yeah, it's really nice what you are saying, but you know, in nature there is a limited number of pigments, and once you get them all, that's it. But if you will work with nanostructures, it was a matter of playing with light and not with pigments. So you can have any color of the rainbow, you can have different effects. And then I was totally fascinated with it. And then I applied for a second part of the project to think about how we can color without using pigments. So since 2020 till now, I've been mainly working on these nanostructures based on a pigment, but still not it's not the pigment what we see, but the light interference. This is a project that we have done with the University of Ghent and 2 American universities about the use of this synthetic melanin. So melanin is the pigment of the skin, but is a material that has some properties that facilitate the creation of structural colors. So for those that are not into color, pigment color is basically a chemical compound that absorb all the colors from the rainbow and then reflect the one that reflect one. So that's the color that we see. And a structural color is a color that is formed because of how light interferes with a structure. So it's the color that we see in a soap bubble, for example. We see a color, but there is not a pigment involved. This is an example of structural colors, but you have to think about this, the butterflies, beetles, birds. Once you are into structural colors, you see structural colors everywhere. But this is an example. So that's what we do in the lab. We make these structural colors based on synthetic melanin by different ways. This, what I showed you here, is something that is called thin film interference. So we create thin films with different thickness. The color, because it's melanin, is brown, but if you find the right angle and a dark background, because it needs a big contrast, you can reveal a color. This is a thin film of melanin on clay, on glass, creating some terrazzo effects, on different type of materials. So basically we coat different materials with this thin film of melanin, and we collaborate with artists. So this research has been quite open already for some time, and from time to time some artists come to the lab to test our materials. And one of the artists is a Veronica Janssens, is a Belgian artist. With her, we had the support of L'Oréal to scale up the research, because also something that that is normally happens is that in the lab everything stays quite small, and the scaling up is always a challenge. And we had this support to do it, and it was really amazing. So yeah, and Veronica is one of the artists that we have been working with, but also we had other artists like with Tina Pikkonen. She's a Finnish artist. She's also creating these big paintings. Because one of the things that the structural colors have, that I didn't say before, is that they can be also iridescent, and because basically the color is dependent on the light interference, they create a kind of, I don't know, need of interaction with the work. So most of the artists that they come to the lab, they are looking for this type of kind of performativity that you have to do in front of a work to experience the whole color. We also have been working with Dimitris. All these works that I'm showing you now, if you are interested in seeing them in real life, from next week on they are gonna be exhibited in Z33 in Hasselt. And yeah, also with we have been coating paper to recreate beetles that here in Belgium are about to extinct on structural colors, creating these big blankets with Helene, where every little feather is colored with structural colors to recreate these feather blankets from the MaJas. And also, yeah, with the jewelry designer, creating also these coatings. As I said, there are many ways of making structural colors. In the lab, we do this thin film interference, but we also make paint by using silica particles and coating them with melanin. For the scientists there, probably you will know, depending on the size of the silica particle, is a color or another. But if you don't, and you need something really dark to see it. If not, it's like a kind of pearl. And we have been testing it on paper. And also we make nanostructures, also with balls, with different size of melanin to create different colors by the light interaction. For me, I mean, doing scientific research in an artistic context is quite interesting because it makes you, go out of your comfort zone. It questions you a lot. I'm now used to it, but at the beginning, when I was starting into this art and science field, I was really surprised how many questions I was receiving, like questions that I never had before. I never questioned myself. So I think that's quite one of the most interesting parts of it. Also, something that is really nice is the unexpected research paths. Also, at the beginning, which I remember when I was at the back, I had these artists and designers coming to the lab and asking me, I would like to do this and that. And then I was trying to help them out, like, if you do this, and giving them protocols or methods. But most of the times they were not following what I was doing. And at first I was kind of disappointed, like, okay, you are coming to me and I give you the method and then you do whatever you want. But somehow, most of the times, the outcomes that they had were much more interesting than the ones that they would have had if they would have to follow my instructions. So that was something that it really fascinated me. And nowadays, much more flexible, as long as it's safe. I'm quite open. It's what I expect, that people hack the methods that we have in the lab. And also, with the color research, it's nice also to work with the main users of my research. So it's kind of nice to extract a pigment or to make a paint or whatever, a dye, and then to have a student or someone else next to me that will immediately try it. And even if it's not working, it's always nice. And and also you were talking about before, like not only painting and not only for painting and drawing. And personally, the outcomes that I like the most are the ones that are not so much connected to this. So somehow it's, as I say, I'm teaching to master students, so I have students from any discipline at CASC. And I know when someone from graphic design comes to the lab, what they are going to do. I mean, I don't know the final outcome, but I know that they will be interested in the colors and they will make some kind of prints or poster or something on magazine or whatever. And it can be super beautiful, but the thing that I like the most is when someone from drama comes to the lab and then something else comes out. And even there is not use of pigments or colors or whatever, but yeah, they come up with these ideas of doing a performance about an algae or whatever, or creating a kind of play about living in the lab, something that I'm not expecting at all. And students from drama or from music that I also have they come with these ideas because for them the practicality of pigments is not so common, and then it's when I think it's when this kind of art and science interaction is super nice. And I think it's also, as I said, in education is for me it's giving access to methods and materials, but also to let them experiment. And as Nicola said, I'm a big fan of art and science collaborations, but the most interesting results is when they come together is on an equal level, not when it's a kind of service like do a nice poster for me with my logo, or can you take a picture with a microscope for me? This is a bit but the only problem that I see is that there are not so much funding for doing this type of collaborations. So most of the times it's based on the goodwill of an artist and a scientist that they are going to work together. But everyone has full agendas. So if there is not funding at the end, it's a bit tricky to follow the collaborations. As I say, for me, working into this art and science is a bit going into the unknown, be open to get any kind of result, and also have a critical approach. I mean, I've been asked so many times why I do need to kill microalgae for extractive pigments, and why the relation between me and the non-humans, the others that are in the lab. That is something that when I was in the pharmacy school, I never thought about it. And nowadays it's part of my work to be conscious about everything. Also the use of other living organisms in my own work. That's it. So let me know if you have some questions.
Yeah, thank you very much. I loved the enthusiasm you had when you said about the connection with other art forms like the drama and the theater. It's really nice to see that it's indeed also going beyond the visual and the painting. I had maybe a question, because with the microalgae, indeed, I was also thinking about how many do you need, but then you said that this project has been stopped. So now is it also a sustainable way of creating, for example, paint or creating color? And is it possible to go beyond and really scale it up for like big purposes, or is there like a limit to the scale up?
It's economically not sustainable at all at this moment, at least not all the colors. You can scale it up, but the only way to scale it up in a way that is kind of sustainable is by doing it in the south of Europe, or in a place where, for example, the energy to grow the algae is not needed. So with the sun is enough. And then in that case, you can scale it up, but not all the algae are nowadays, and maybe in the future, are sustainable. The production and extraction and everything, it is, but at the end it's an expensive product, and also not very stable product. It's something that I didn't say, but not all the pigments from the microalgae, but most of them are chlorophylls, carotenes, phycocyanidins. They are not, if you compare it with a normal pigment, with a synthetic pigment, it's not stable at all. But luckily for me and for the world, more and more the students are not so much interested in creating something that stays there forever, and they are quite open to experiment with other types of materials that eventually can fade, and that's it. And even thinking on these properties as a good thing, like, okay, I can print a newspaper or a poster, and in 6 months reuse the paper again and print something else on top, and that's it.
And the current project, the nanoparticles, is that like a sustainable and scalable?
Again, economically, it's not sustainable. I will say it's an expensive procedure, but I think in that case it can be scalable because our partner, one of the partners in the States, in Chicago, they're already working on scaling up the production of this synthetic melanin, not for coloring, not for playing with the light interaction of the particles, but for medical reasons. I think they are planning to scale it up, yes.
Okay, nice. Somebody in the chat also asks to share more information about the exhibition in Hasselt. I think, sorry, there will also be a link probably that I will share in the follow-up mail, but maybe if you can already put it in the chat, then people can find information. Someone else suggests, I think it's just for your research, whether you've tried to culture them on high temperatures, like up to 1,000 degrees. Maybe it's a nice thing to keep in mind. I maybe also had to end because we're coming to the end of the talk and I don't want to keep people. Maybe one last question. What do you think is the biggest hurdle to build the bridges between art and science? And I think it's a question to everyone, to all of our speakers. If you would say like the biggest hurdle you encountered, or that is maybe more interesting currently, that you think is the biggest roadblock between really connecting art and science, what would it be?
Okay. I think it's money.
Money?
Yeah, I think so. As I said, now many projects, art and science projects, are based on goodwill, and this is not sustainable in the long term. So it should be, and there is some funding for art science project, but still is quite little, and yeah. And also accepting that, because accepting that this type of projects might end up with nothing. So the how
the result might not be what you foresee, but that's true for all science. I think that should be more.
Yes, but normally when you are writing a scientific project, you have an idea about what you are gonna get, and maybe you even got it, and then you are applying for it. So that's something else. But with this art and science, I think the beautiful thing of it is applying for something that is not clear. That is open enough to play with it, and maybe it's not what expected, but maybe it's even better. So yeah,
maybe it's a call for funding or funding agencies to consider it as well. Any other?
I agree with Maria. Funding is very important, and also the validation of artistic research into sciences, and to get that freedom indeed to experiment. And I think it's quite important, you know, to have the freedom and the time to do that. And for that, you need some funding, because we can't live off the air, you know.
Anne or Nicola, do you have any additions, or is it also what you feel is the biggest struggle?
No, I can add something. I think looking back into the past, it's not a real struggle, but you see that mostly the scientist is recognized and the artist name is not always mentioned. And I come back to Vesalius, for example. Everybody knows his work, and we almost assume that he made all his drawings and graphics and interpretations, but it was an artist who also performed maybe the dissections, maybe was actively involved. And I hope it will be a lesson. We should recognize each other in scientific literature. Sometimes people they don't mention the name of the artist already. So it's the written word is still the most recognized scientific word, and there are so many other languages. So I think that's a hurdle to overcome, because these written thing is very easy to handle, and it's also very easy to have it in laws. It's very easy. But another language like the drama, as Maria said, maybe it's much more difficult to have that validated.
It's good to spread awareness about it as well. Yeah.
I'm not fully agree. Sometimes I see artworks that the one that is not named is the scientist behind it.
Okay.
But yeah, I think because nowadays, I mean, especially if you are a well-known artist, you just can just take it and then the science behind is a bit.
So it works both ways.
Yeah, I think so. I think so. Yeah.
So we need appreciation from two sides, actually. Nicolas, any final?
Yes, I want to add, but MaJa also mentioned it already. I think it's also the busy agendas everybody has, and that we also live, I think, also in the science and in the art in a very specialized world. And you have to break it open again and make it not as clear as it is right now if you want to do this research. And then, of course, because of the fundings and because of everything, because the target is not clear, it's more difficult to start with it. But I think it's a little bit what MaJa also said. But I think that I feel in the Jonge Academy that this is quite problematic when you have a group and it's a group of scientists, it's scientific topics, and then as an artist, you're just sitting there and you can't add really anything. And the other way around, when we have something that is more specialized in the art, then scientists, of course, they help us, but it always stays like a little bit of a sidekick. So the really merging together is difficult to get. But of course, we have to keep fighting for that.
Yeah, hopefully this lunch talk can help a bit to do that, and all of the links and everything and all of the initiatives that have been shared in the chat. So I will, for sure, for everybody that's still listening, I will share all of these links also in the follow-up mail. So if you didn't get the chance yet to visit them all, I will share them later on. And with that, I think we're at the end of our talk. And I, of course, want to thank all of the speakers for sharing their insights, their expertise, answering the questions. I thought it was really interesting for myself as well, because I'm a scientist and I got to get to know a lot of things today. So thank you again from my side. And thank you, everyone, for being here and for listening. And I hope to see you in our next lunch talk on May 16th. And have a good afternoon and a sunny day. See you soon. Bye-bye.
Thank you.
Goodbye.